Hello, we're going to go ahead and get started. Thank you everybody for coming to Drosophila Models of Human Disease number 1. My name is Rebecca Kripke, and I am here from the University of Washington, and I am very excited to introduce our first speaker, who is going to be Bess Frost.
So I'm going to be talking about Alzheimer's disease today. Do I have any Alzheimer's people out there? All right, nice. Okay. Okay, so we've known about Alzheimer's disease. Next slide. I was warned about the stalling. Next slide, please. I'm using the arrow key. Thank you. So this was work that I performed as a postdoc in Mel Feeney's lab. It just came out, I just published it in January and started my new faculty position at the Barshop Institute in San Antonio and UT Health Science Center. I will keep pushing the arrow. Yeah, I don't think it's user error. Okay, so we've known about Alzheimer's disease for over a century now. It was first described by Alwa Alzheimer in the early 1900s in the brain of his former patient named Augusta Dieter. After Augusta Dieter died, Alzheimer did an autopsy on her brain and saw what he described as plaques and tangles. We now know that the tangles are composed primarily of a protein called tau, and the plaques are composed of amyloid beta. So tau is most well known for its role in Alzheimer's disease alongside amyloid beta. However, tau is the primary pathological feature in a handful of other neurodegenerative diseases in the absence of amyloid beta. And we know that tau dysfunction is sufficient to cause disease because mutations in the tau gene are associated with familial tauopathies. So the talk I'm going to tell you about, my work I'm going to tell you about today, is an extension of some previous work that I had done where I used a tau transgenic fly model, a mouse model of, the arrows are working now, thank you. Okay, there you go. Where I had used a tau transgenic fly model, a tau transgenic mouse model, and human Alzheimer's disease brains to show that pathological tau causes a widespread relaxation of constitutive heterochromatin, and this allows genes that are normally embedded in constitutive heterochromatin and silenced by it to become expressed, which activates the cell cycle in neurons, which are normally postmitotic, and it's well known that activation of a cell cycle in neurons is sufficient to cause their death. Okay, so when I came to this project, my next question was, how is pathological tau causing heterochromatin to relax? And I became really interested in a group of disorders called Laminopathies. These are due to mutations in the Lamin gene. And the reason that I was attracted to them was that a well-known feature of Laminopathies is relaxation of heterochromatin, what I had just seen for tauopathy. So the Lamin protein is a structural protein of the nucleus. It forms the nucleoskeleton. It coats the internal surface of the nuclear envelope and provides attachment sites for heterochromatin by a direct interaction between Lamin and heterochromatin protein 1. So in these diseases, when you have dysfunctional Lamin, the heterochromatin detaches from the Lamin nucleoskeleton and relaxes. One phenotype that can result from Lamin mutation is a progeroid syndrome, which is a premature aging syndrome, where young children have aspects of aging starting around the age of 6 or 7. An interesting aspect of these diseases is that many tissues are affected, but there's no neurodegeneration. And of course, for most neurodegenerative disorders, aging is the greatest risk factor. So this is a big question in aging research. Why is there no neurodegeneration in these kids? And it led some to hypothesize that the brain is not susceptible to the same basic mechanisms of aging as other tissues in the body. So despite the fact that there was no neurodegeneration in these kids, I went ahead full steam ahead with my hypothesis, which was that tau causes a Lamin dysfunction, which is upstream of the heterochromatin relaxation, which is the cause of the heterochromatin relaxation in Alzheimer's disease and associated tauopathies. So I took a look at postmortem brain tissue from control and Alzheimer's disease brains, and I saw that in the healthy controls, the Lamin is in red, nuclear pores are in green, that the nuclei were pretty nice and spherical. In the Alzheimer's disease brains, however, there's a significant disruption of the Lamin nucleoskeleton, where I could see the nucleoskeleton coming in from the periphery into the interior of the nucleus. And I have a co-stain here of nuclear pores because I want to show you that it's not something that's just specific to the Lamin nucleoskeleton. I think that it involves the entire nuclear envelope. You can see this in the video here. Slow it down. On the right is the Alzheimer's is a nucleus from an Alzheimer's disease neuron, and you can see the tunnels coming in to the nuclear interior. I see this in about 60% of neurons in the Alzheimer's disease brains. I only see it in about 20% of the neurons in control brains. When I look at overall levels of Lamin protein by immunofluorescence, I see a slight but significant decrease in the total levels of Lamin protein based on immunofluorescence. So the Alzheimer's disease brain here, we're looking at total Lamin B fluorescence intensity. You can see that it's shifted downward compared to the control brains. All right, so does this matter? Does this dysfunction of the Lamin nucleoskeleton actually have any consequences for neuronal health and survival? I turned to the fly model of tauopathy. This was first developed in 2001 by Mel Feeney, my postdoctoral mentor. It expresses human tau specifically in neurons using LEV-GAL4. They have progressive neurodegeneration in adult flies. They have a shortened lifespan and health span. And importantly, many critical aspects of human tauopathy are conserved in this model. So just taking a look at their brains at 10 days old, all the experiments that I'm showing you are in 10-day-old flies. Control flies have this nice spherical nucleus, just like I see in the human brain. However, in the tau transgenic flies, I see that the Lamin comes in, just like what I saw in the Alzheimer's disease Alzheimer's disease human brain. This is happening in neurons based on co-staining with LEV. And in the flies at 10 days old, it's about 15% of neurons that have these Lamin invaginations. Overall levels of Lamin protein are also decreased in the tau transgenic fly head. So you can see the control Lamin levels here, and then a significant reduction in the tau transgenic flies, suggesting that not only is the Lamin changing its location, but the protein levels are actually decreasing. All right, I then combined the tau transgenic fly with a further knockdown of Lamin using 2 non-overlapping RNAi lines, and I looked at neuronal death based on tunnel staining in the brain at 10 days. So tau expressed alone has a significant amount of neuronal death, and then when you combine that with a further reduction of Lamin, I saw a significant increase in neuronal death. Now, of course, I tried to overexpress Lamin and reverse this. Unfortunately, the Lamin overexpressors were toxic on their own in the brain, so I couldn't use those modifiers, but I'll show you in a few slides how I got around that problem. Okay, so at this point, I was pretty convinced that pathological tau can drive Lamin dysfunction and reduction of Lamin proteins. And so I wanted to know if Lamin dysfunction could be the reason that heterochromatin is relaxing and I'm getting all these other downstream events, because previously it was thought that in the adult brain, Lamin might not matter as much as it does in other tissues. Okay, so taking a look at heterochromatin. In neurons based on LEV staining, control flies this is a heterochromatin based on heterochromatin protein 1 staining. They have a lot of heterochromatin in the nucleus. You can see these HP1 positive chromocenters here in almost all of the nuclei. In tau transgenic flies, you see an overall reduction of heterochromatin protein 1 staining, and you lose that chromocenter staining as well. This is a Lamin allele that was developed by Janice Fisher. I'm using it as a homozygote. It has a removal of the domain that's responsible for targeting Lamin to the nuclear envelope, so it's a loss of function. And it looks just like the tau transgenic fly, suggesting that Lamin, and this is in the absence of tau for the Lamin A25. So this suggests that just like in laminopathies in humans, Lamin dysfunction in neurons is sufficient to cause heterochromatin relaxation. All right, I showed you previously that the heterochromatin relaxation causes neurons to reactivate the cell cycle, and this causes their death. In this slide, we're looking at cell cycle activation based on proliferating cell nuclear antigen. In controls, in the neurons, there's no cell cycle activation. In tau transgenic flies, you have pCNA staining here, and then I also see it with the Lamin loss of function. Dysfunctional Lamin also causes neuronal death based on tunnel staining. So in the tau transgenic fly, you see the nice tunnel staining here, and then I also see it in the Lamin, clearly in the Lamin mutant, clearly showing that Lamin dysfunction has serious consequences for neurons. All right, so the next question is, how is pathological tau causing this dysfunction in Lamin? It had already been shown that tau causes an overstabilization of filamentous actin, and I thought that this could perhaps be the connector between tau and Lamin dysfunction. So the actin cytoskeleton interacts with the Lamin nucleoskeleton through a complex called the LINC complex. The LINC complex spans the nuclear envelope. It's conserved from flies to humans. It has an outer protein component and an inner protein component in flies. This is MSP300 on the outside and COI on the inside. So I thought that tau-induced stabilization of filamentous actin might affect this LINC complex in some way, which would then cause this dysfunction in Lamin. So I drove filamentous actin stabilization by tau, or by overexpression of WASP, or by overexpression of SPIRE. And I took a look at the LINC complex based on staining with COI. That's the component that's on the inner nuclear envelope. In controls, COI circles the nuclear envelope, kind of appears similar to Lamin. However, in the tau transgenic flies, I see a clustering of COI, suggesting that the LINC complex is clustering at one part of the nuclear envelope. I see the same thing when actin is stabilized by a pure actin stabilizer. I see this clustering, and I also see it with overexpression of UAS SPIRE. All right, so I told you before that I wasn't able to just overexpress Lamin in order to suppress tau toxicity because Lamin overexpression was toxic in the brain on its own. This didn't come as a huge surprise. Duplications in Lamin cause a human disorder, so I suspected that Lamin overexpression would be toxic in the first place. Anyway, I wanted to see if I could manipulate this pathway and suppress tau toxicity. So I reasoned that if this whole pathway is being kicked off by filamentous actin, that reducing the interaction between filamentous actin and the LINC complex might nullify the role that this overstabilized actin was having. So I combined the tau transgenic flies with with RNAi of MSP300, and I saw that Lamin levels came back up. And what happens when those Lamin levels come back up? I see less neuronal death in the tau transgenic flies, suggesting that filamentous actin is affecting the LINC complex, and this is affecting the Lamin nucleoskeleton. All right, it's not time yet, right? I'm good? Okay, so at the end of the day, the take-home, I've only showed you a snippet of data from the paper, but at the end of the day, I hope I've convinced you that Lamin dysfunction in the brain has significant consequences, and that Alzheimer's disease and associated tauopathies are actually acquired neurodegenerative laminopathies. I'd like to acknowledge the brain donors and their families. This work, as I said, was done in Melfini's lab. Farah Bardai is a co-author on the paper. I didn't talk about her contribution. I didn't talk about any of the things that she contributed to. This is my own lab now. They didn't help out at all, but I'm very excited to have a lab. So I put them up there. Thank you for all of the fly contributions at all the work people do, the Vienna Stock Center, the Trip Stock Center, Neuromab, the Hybridoma Bank. This was funded by a pilot grant from the ADRC and the Harvard Neurodiscovery Center and a K99. This is my lab now. I'm recruiting postdocs, so if you're interested, please email me.